a powerhouse Wahine finish us off for the day. So I would like to introduce Lauren Smith with monitoring Culex quinquefasciatus in Kīpahulu Valley, Haleakalā National Park to support future landscape mosquito control. Take it away, Lauren.
Smaller than two people. Aloha kako. My name is Lauren Smith, and I'm a biological science technician, wildlife, for the U.S. Geological Survey Pacific Island Ecosystems Research Center, or USGS PIERCE. Today, I will be presenting our ongoing project monitoring Culex quinquefasciatus in Kīpahulu Valley, Haleakalā National Park, to support future landscape-scale mosquito control. I'd like to start by acknowledging the aina on which we gather and on which this research was conducted as an indigenous space, and it is recognized by Kanaka Maoli as our ancestral grandmother, Papa Hanomoku. For thousands of years, the Hawaiian honeycreeper has spread across the archipelago, adapting and splitting into a large radiation of species. In 1826, the southern house mosquito, Culex quinquefasciatus, arrived to the islands in the ballast of ships. Shortly after came a parasite, a plasmodium that causes avian malaria, a novel disease for which Hawaiian honeycreepers have little to no resistance. As we speak today, 3/4 of all honeycreeper species have gone extinct, and we are on the verge of losing 4 more. To combat avian malaria and slow the wave of extinction, the National Park Service and its partners are preparing to use the incompatible insect technique, or IIT. This process involves flooding the wild Culex population with males, lab-bred to have a different strain of the gut bacteria Wolbachia. If a male Culex has a different strain of Wolbachia than the female he mates with, the resulting eggs will not be fertile. With sustained releases, this method can suppress the wild Culex population. Home to the Maui endemic endangered kiwikiu and akohekohe, Kīpahulu Valley is a priority site for IIT release. Before we can release males, however, we need to understand the baseline demographics for the Culex population, as well as prepare for monitoring after the release. USGS PIERCE was tasked with finding this information. The first thing we want to know is the density of the Culex population at both high and low elevations, as well as seasonal variations in that density. We want to find where the Culex are breeding and how those elevational populations are related. Past studies in other locations around Hawaii have shown low densities of mosquitoes in native rainforest. So we also wanted to figure out which types of mosquito trap are the most efficient in these conditions. And finally, we wanted to develop a protocol for using environmental DNA, or eDNA, to more easily estimate density and abundance without a large-scale trapping effort. To answer our questions about demography, population genetics, and trap efficacy, we selected 3 types of traps to capture adult mosquitoes. All of these traps use lures to draw them in and a fan powered by either a 12 or 6-volt battery to create a suction that pulls them into a net bag. The Biogen Sentinel and Pro traps both use carbon dioxide as their main lure and attract female mosquitoes who are looking for a blood meal. The Sentinel also uses a chemical attractant that smells like your worst stinky gym socks. As you can see, sentinels sit on the ground and must be covered by tarps to keep the constant rain from blocking the suctioning airflow exchange system. This is the main trap used by conservation and research agencies across the state. Biogen's Pros, however, are a new trap that functions very similarly, but runs off a smaller battery and thus can be hoisted into trees to get closer to the birds. Depending on the field conditions, we have hung them between 10 and 15 feet in the canopy. The last type of trap we are using in this study is called a rabbit trap. It uses stinky water that is fermented with hay and yeast to make it more attractive to females looking for a place to lay their eggs. Using both carbon dioxide and infusion traps informs us about the life stages of the adult mosquitoes in our trapping site. Because mosquito larvae are aquatic, to answer questions about larval presence, we have to turn to streams and pig wallows. We divided the accessible areas of major streams into 10-meter sections, from waterfall to waterfall. In each 10-meter section, we use mosquito dippers to sample 10 times in 10 different pools of non-running water. When we find larvae, we then dip that pool a total of 10 times and record the number and age of the larvae in each dip to estimate the density of larvae present in each pool. We then also take a sample of the water and force it through a DNA binding filter. eDNA is a technique that uses tiny particles of DNA shed by all organisms to test for presence of a specific organism. We are still in the research and development stage of this technique in Hawaii and have tested 4 different collection devices and filters in the field so far. For this study, we selected 2 camps on the lower shelf of Kīpahulu Valley, Delta, the first picture, at 3,000 feet, and Polychaea, the second, at 4,200 feet. To achieve a random, even sample across each site, we alternately placed 15 sentinels and 15 pros 100 meters apart. Because gravid traps attract mosquitoes in a different life phase, we chose to pair a gravid trap with each pro trap, for a total of 45 traps at each site. On these maps, teal teardrops are the stations with the paired pro gravid traps, and purple teardrops are the sentinel stations. Due to the terrain and the location of trails, the trapping grid at Delta covers a total of 780 feet of elevation, while the trapping grid at Polychaea only covers 440 feet. Together, the trapping grids at both field sites cover 1,720 feet of elevation in Kīpahulu. To understand the seasonal cycles of Culex quinquefasciatus, we use previous studies to break the year into 4 seasons. The population peaks in August through October, decreases November through January, is at its lowest February through April, and increases again May through July. We had planned to sample each site twice in each of the 4 seasons, but logistically this proved infeasible. Helicopter flights, financial concerns, and weather conditions all make this type of monitoring work very difficult, and the lessons learned from this project will help inform the planning of future mosquito monitoring. At this point, we have trapped at Delta 4 times, beginning in August, and at Polychaea 3 times, beginning in September, for a total of 7 trapping trips. We hope to visit each site 3 more times to get a baseline for the trough and increasing seasons, and 1 more data point for the peak season. As this is an ongoing project, all data at this point are preliminary, but we are seeing some interesting patterns. As we expected, we are seeing many more mosquitoes at the lower elevation site than the higher. Over the past 8 months, we have caught 356 Culex at Delta and only 10 at Polychaea. To quantify this data, we divide the number of mosquitoes captured by the number of nights spent trapping to get the average mosquitoes captured per trap night. Those numbers are plotted on the y-axis in both of these figures. On the left, we have mosquitoes per trap night plotted against the trap type at each camp. At Delta, with a higher sample size, the pros, seen here in blue, are outcompeting both of the other trap types. At Polychaea, only 2 sentinels have caught 8 out of the 10 mosquitoes, so with such a small sample size, this could be due to the individual trap locations. The figure on the right shows us the seasonality of our results for each site. To account for daily variations, we took an average date for each trip and plotted the whole trip's capture rate on that point. Early fall was indeed the peak at each site, and March was the trough for Delta. January shows a small peak at both, but I suspect this may be due to the weather patterns Kīpahulu experienced that month. To date, we have dipped 1,050 meters of streams at Delta and 700 meters of streams at Polychaea on each trip. At Delta, we found more than 30 pools with another mosquito species, Aedes japonicus, and only 4 pools of Culex larvae. At Polychaea, we found 6 pools of Aedes and 0 Culex. Almost all of these pools are located in streams, but we did find a pig wallow with Culex larvae present from August through January and not present this month. Culex prefer mud and detritus-rich substrates, such as the pig wallow in the upper right-hand corner. Aedes prefer rock pools, such as the picture to the left of that. So it's not surprising that we find more Aedes in the stream beds. The detritus in the pools is our biggest issue when it comes to collecting eDNA samples. The filters are extremely small and get clogged very quickly, limiting the amount of water we can sample. The hand pump has been able to filter the most water by far, including 50 milliliters at the pig wallow. The Stairvex is next, and the syringe with the 2 other filter types filter the least, with Natrix Q able to handle only 3 milliliters of water from the pig wallow. We have not been able to process the DNA in the lab yet, so we'll discover how efficient each type is at binding DNA soon. And as this is an ongoing study, there are many more strands of this ecological web to uncover. We hope that the results of this study will help inform the IIT release efforts and make Kīpahulu Valley a place the Manu call home for many years to come. Mahalo. And mahalo to all of our partners and our 15 volunteers that have helped in the field. We can't have done this without you. Any questions?
With the eDNA collection, you have a process here. How are you storing it in a picture?
So we're putting it in different types of buffers depending on the type of collection system.
Could you possibly bring filter water that has a lot of to try to get in balance so you can get more column? Because the eDNA it should slow pass through the larger filter first, and then you can.
Yeah, I think we're a little worried that the DNA might get stuck in the, like, Pigwalas have just so many pieces of mud in it. I think we're a little worried that if the mud gets stuck on the filter, it's preventing some of the eDNA.
I agree that could be a concern, but if I mean these assays can be fairly sensitive. I think that like collecting more, even if it passed through a filter previously, could improve saving useful return. Something to consider.
Yeah, absolutely. Yeah, thank you.
Haleakalā was like,
Mhm.
so there's another camp. I'm seeing
So on the lower shelf, oh, sorry, the question, I forgot to respond or to repeat that. But the question is there another camp higher than Haleakalā? So on the lower shelf, Haleakalā is the highest camp. On the upper shelf, however, there are higher camps, but we see so few mosquitoes at Haleakalā that going to any of the other camps probably would just be a bunch of zeros, which is important data. But yeah, so he just asked if there's any hapu'u cavities that we're finding them in. So on the Big Island, we have a lot more hapu'u, and so there's a lot more cavities, and we're finding mosquito larvae there. But on Maui, especially in Kīpahulu, we really don't have that many hapu'u. I found a single cavity at Haleakalā, and so we've collected eDNA samples from that, but it has never had Culex or any type of larvae so far. Yeah, so he asked if we found any 80s or Culex in the same cavity. So far, no. We are keeping an eye out for them. They are relatively easy to identify in the field, unless they're like the very like baby and star larvae. We are collecting all of them and bringing them back for more identification in the lab. And so far, we've only seen one of each type in each. Any other questions?